Assalamu alaikum everyone. This is Kazi Jannatun Nayeem. Today my presentation topic is unveiling the phytochemical and pharmacological potential of Amora rohituka seeds and food pulp in vitro and in vivo approach. Here is my content: introduction, literature review, plan profile, aim of the work, methodology, result and discussion, and conclusion. Bhaiya, am I audible?
জি শোনা যাচ্ছে।
Okay. Here is my introduction part. This part I will describe about medicinal plant. Actually, we know that medicinal plant is any plant that contain bioactive compound that can be used to prevent, treat, or cure disease in human or animals. These plants have been traditionally used in herbal medicine and modern pharmacology to produce natural remedies, supplement, and pharmaceutical drugs, etc. According to Bangladesh Agriculture Research Institute, BARI, they are discovered 722 species of medicinal plant in Bangladesh, and it is estimated that 70 to 95% of the population in developing country continues to use this traditional medicine even today. Here is one of medicinal plant I mentioned is Bacopa monera. One of medicinal constitute bacoside. Other constitute have remained on this compound. I only described and mentioned on this compound bacoside. It's generally used memory improvement, insomnia, epilepsy, and as an anxiolytic, attentive deficiency, hyperactivity disorder. It's a short term we describe ADHD. This is the literature review of the my medicinal plant, which is Amora rohituka, and this literature review describes that previous activity of the other researchers conduct, and here is the paper link. And they conduct some like antioxidant, cytotoxicity, antimicrobial activity, methanol extract. It's different soluble partition of the bark, leaves, and Amora rohituka. They describe about the BBPH method of crude methanol extract, bark, and chloroform soluble fraction and crude extract. Other paper we found that Amora rohituka, they evaluate the anti-inflammatory effect, and other inhibition that microbiological activity has remained or not. Besides, inflammation is critically incremented degenerative CNS pathological, including Alzheimer's disease, and it's also known as accelerated and disease outcome. It's also Amora rohituka plant evaluate that this chemical compound and remain on this plant. Some other literature review also remain. I discovered about 301 of them. Here is the plant profile. I generally worked on the fruits, seeds, and leaf. Other part also discovered on the other author. And here I mention about the literature review. Here is the literature review taxonomic classification. My aim of the work was the estimate the phytochemical and pharmacological activity, antioxidant, analgesic, neuropharmacological potential of Amora rohituka to determine if it can be good source of the future drug and development. Some specific objectives are like evaluate the qualitative and quantitative screening and determine the in vitro and antioxidant activity of crude methanolic extract by DPPH and ABTS radical scavenging method. This is also antioxidant activity. I evaluate through this method DPPH, ABTS. It can also be evaluated by peroxide method. I didn't mention here. I only describe about DPPH and ABTS. Also, I analyzed that in vivo method, analgesic activity of seeds and fruit pulp extract by hot plate method, central analgesic test, and acetic acid induced breathing test. Also, I evaluate the neuropharmacological activity of the crude methanolic extract of 𝐜𝐺𝐻𝐿. Now I discuss about methodology of this research. This is the extraction process, how I extract this plant. Firstly, I collect the sample. This is the fruit, my medicinal plants, fruits. Fruits. Then I dry this sample, and after the drying, I grinding through this method, and this powder form I soak in the methanolic solution for the 72 hours to it's more than more it can be kept on the 7 days. 7 days. Generally people kept 3 days to 4 days. I also kept it 72 hours. After that I filtered this compound, and when it's evaporated, I mm, I took it on the ampule. This is the fruit pulp, and this is the seeds extract. Within two extract, I conduct my whole research. Some qualitative test I evaluate through this method, like cardiac glycoside, alkaloid, protein, steroid, tannin, phenol, flavonoids. Cardiac glycoside I evaluated through this test, like Keller-Killeney test, and this result come positive. Is double it is a triple plus means it's more prominent than seeds. Food plot if food plot extract should more prominent than seeds. Then alkaloid test I evaluate through the Wagner test, Hager test, Meyer test. We can observe that result of seeds here triple plus, that is 1 plus. That is determined that seed extract more prominent than fruit pulp. This is also happened like Hager test, Meyer test. Then protein secondary metabolite I evaluated through the BIIHR test, steroid test, Swarovski test, and Lieberman-Brooker test, tannin, perichloride, phenol, perichloride test, flavonoid hydrochloride test. Some of the figure I mentioned color, how color came out after the result of after the through this test procedure, like glycoside, cardiac glycoside, alkaloid Meyer test, alkaloid Wagner test, tannin, and steroid. TPC. TPC হচ্ছে total phenolic content, এটা TFC হচ্ছে total flavonoid content, TTC হচ্ছে total tannin content. Here I use for the TPC I use gallic acid as a standard cup. Then for the TFC I use as a standard cup quercetin standard cup. And TTC হচ্ছে tannin, total tannin content, and I as a standard I use tannic acid. This is the result: food pulp 38.17, seeds 22.14. This result came out comparison of the standard of gallic acid curve and TFC. This is food pulp result came out like 14.1 and seeds 9.34. This result also mentioned here comparison to quercetin standard curve, as well as tannic acid and total tannin content mm, and compared to the standard curve of tannic acid. Here food pulp 8.12 and seeds to 2.21. Now I describe about antioxidant activity. Here I mentioned three methods. I conduct the my extract through these three methods to evaluate the antioxidant activity is remain or not. This is the DPPS free radical scavenging activity. This is ABTS free radical scavenging activity. I will not describe the whole method and but I overview some of result content how it come. Like in the DPPS free radical scavenging assay, extract solution I preferred. Then another way I DPPS solution prepared another beaker. Then after the hour, after the certain hour, I add the DPPS solution and kept it incubation for 1 hour. After adding DPPS extra solution extract, I kept it incubation for 1 hour. Then after the incubation, I measured this UV absorbance through the UV method. It's a wavelength 517 nanometer. Then like ABTS free radical scavenging assay, the extract solution same as well as for like DPPH. Then I preferred ABTH solution. This is ABTH solution. Then then after this adding the after this prepare the ABTH solution, I add this solution through the extract solution. Adding ABTS to the extract solution, I measured the absorbance at the 734 nanometer. As well as hydrogen peroxide scavenging activity, I evaluate on this extract. Here the prepared extract solution. This is the my hydrogen peroxide in phosphate buffer saline. And through the certain process, I add this solution on this extract. After 10 minutes later, I measured this absorbance at 230 nanometer. In DPPH scavenging activity, I measured through the standard of ascorbic acid. Here, graphical presentation is mentioned here, like food pulp seeds. Mm. In DPPS study scavenging activity, extract short concentration dependent antioxidant activity. This higher percent of scavenging activity was found at concentration of the 1000 per milligram per ml. The extract of the food pulp seeds showed moderate antioxidant activity compared to the standard cup. Then I will describe about the ABTS scavenging activity. This also this extract also showed the concentration dependent antioxidant activity. This higher percent scavenging activity was found at the concentration of the 1000 per milligram per ml, and then the extract of food pulp of seed showed moderate antioxidant activity compared to the standard cup. Here I mentioned there this is the concentration dependent antioxidant activity. It's like we observed that firstly it was 56.74, then 65.40, then 96. And 96.9.08. When we increase the concentration, it also the percent of scavenging activity is also increased. That means it's the concentration dependent scavenging activity. It's as well as for the seeds. This is all the hydrogen peroxide scavenging activity. In the hydrogen peroxide scavenging activity, extract short concentration dependent antioxidant activity like DPPH, DPPH and ABTS method. The higher percent of scavenging activity was found at the concentration of as well as 1000 per milligram per ml. The extract of the food pulp seed showed moderate antioxidant activity and compared to the standard cup of ascorbic acid. This is the gradually increasing like 45.50, 52.38, 85.56 compared to the ascorbic acid standard cup. Then I compared the three method, the DPPH activity, ABTS activity and hydrogen peroxide activity. For this method, we observed that in the ABTS method, seed shows more prominent activity than other method like DPPH and hydrogen peroxide. Another way in hydrogen peroxide method, we observed that food for showed that more prominent activity than any other method like DPPH and ABTS. His prominent activity like 300 to 1.41. Then we discover the analgesic activity. Mm, Analgesic activity. I evaluate this activity through these three method like hot plate method, acetic acid reading method, and center analgesic activity. Mm, this is the huge. Sorry. Mm. This is the huge time to take to elaborate. I overview the within short term like in hot plate method we firstly we marking the mice. You can see red color, through the red color I mark this mice. Then I prepared the extract, then extract the certain solution, solution 200 for 200 milligram per ml and 400 milligram per ml concentration. Then I feed this extract to the mice. Then I put the mice on the hot. This is the hot plate method. Then I put the mice on the hot plate method. Here I set the temperature 55 degrees Celsius. Here I observe that how within how much time the mice jump and how many time it can sustain on this hot plate method. This is the hot plate method. This is in this you can see this base to reach the heat. We observe that how much time these mice can sustain on this. Then acetic acid reading method. Firstly, we mark these mice. Then after we feed the mice, then and acetic acid we and peripheral way we induce this acetic acid and we observe that how many times that these mice can read this his arm. Then after that center analysis activity, we as well as we mark this mice and after then we feed these mice. Then this is the hot water. Hot water. Hot water. Here we observe that mice tell how much time it can sustain on this hot water. Then after this, a three method, hot plate method, acetic acid reading method, and central analytic test or method. We observed that we observed that this is the standard cup. Footfall content basal reaction time content 1.889 and for the concentration of 400 2.75. This is also concentration dependent manner. This is the seeds 1.51 for the 400 1.81. This explain that this explain that within the concentration, if we increase the concentration, its basal reaction time also increased. Then acetic acid reading method. This is the standard curve. This is the footfall for the 200 concentration. Is a footfall 400 concentration. This is the seeds for 200, seeds for 400. This is the center analgesic test. This standard cup, we can observe that this center analgesic test we conduct within 0 minute, 30 minutes, 60 minutes, 90 minutes, 120 minutes. This about the hot plate method, acetic acid reading method, center analgesic method. We can discover it elaborately because of short time period. I overview this just like putting shortly. Then my main research about in vivo neuropharmacological evolution test. Neuropharmacological evolution test can conduct several methods like open field, whole course, whole board, EPM, এটাকেও আবার elevated plasma test, light drug and YMS and TMS. This I will describe about four methods like open field method, whole cost, EPM, dark light. Um, in open field method, generally we observe number of square cross by experimental mice for 5 minutes at the dose of 200 400 milligram per kg, the compared to the diazepam at the dose of 1 milligram per kg. We how many square? How many we put the mice and mm, how many square in the mice passed within 0 minute, 30 minute, 60 minute, 120 minute. We count this. We count this simultaneously. This is the open pill apparatus. Mhm. Here, we generally estimate the locomotor activity of the mice and hyperactivity and exploration behavior of the mice. We conduct this method and evaluate this 0 minute, 30 minute, 60 minute, 90 minute, 120 minute. Here is the my result for the open pill test. This is the control. Here has no extract and diazepam for the standard and footfall 200 for the concentration, 200 footfall for the 400, seeds for 200 seed concentration for 400. We can observe that in open field sedative study, we have observed that is dose dependent sedative activity for both food, pulp and seed Amora rihuteca. Generally, seeds extract showed higher sedative activity by controlling locomotor movement of the mice at the dose of 400 milligram per kg compared to the standard. Here standard we used diazepam. Then we discover the whole course test. This is the whole course apparatus. In this whole course study, we had observed number of passes on this apparatus. Here remain in the hole. This is clearly not showed here. This picture, this hole. Through this hole, mice passes, and how many times each passes would estimate this within 30 minutes, 0 minute, 30 minutes, 60 minutes, 90 minutes, and 120 minutes. How many times the mice passes this hole through this apparatus? This is the whole cost test. As well as a standard, we use diazepam for the standard. Then in whole course is generally estimate for the antidepressant activity. Whole course test, we have observed the dose dependent sedative activity for both fruit pulp and seed Amora rohituka, and seed extract should also higher sedative activity by controlling locomotor movement of the mice at the dose of 400 milligram per compared to the diazepam. We can observe 0 minute, 30 minutes, 60 minutes, 90 minutes, 120 minutes. How many times it passes? Then check EPM. This is also called elevated plasmase test. Firstly, I mark the mice and feed this extract. Then I observe this anxiety activity through this apparatus. This is the we can saw that this is the apparatus. This called ophanum. This is called chromosome. How much time remain on the ophanum and how much time remain on the chromosome? We estimate this through this method. And then we observe that if we put the mice on the central, then we observe that if the mice enter the chromosome, then how much time it remain, we count it as a through the stopwatch. Then if it came out from the chromosome, then we started counting for the ophanum. This is called ophanum. This is called chromosome. As a standard, we also use diazepam. Then footfall 200 concentration, footfall for 400 concentration, graphical presentation. For the seeds 200 and seed as well as 400. We can observe that here this golden color remain number of entry in ophanum. I mentioned here ophanum called, this is ophanum, and this is the black one is the chromosome. And time is spent in the ophanum. We mark the green color, and number of entry ophanum in we mark is a golden. We can observe their footfall count 18.16 and 46.66 is for the number of entry. Then another test is light drug test. This is this is the apparatus for the light drug test. Here the drug portion remain on this. I covered the through the board, and this is the light portion. I put the mice on this portion on this part, and I counted that how many time it entered the drug portion and remain on the drug portion, and how much time it remain on the light portion. This is the graphical presentation for the light drug test. This is the control. Here is no extract. All other chemical compound is remain on this, and this is the standard. This is the footfall. I count this 0 minute and 60 minute. In the previous one, you can observe that I mentioned 0 minute, 30 minutes, 60 minute, 120 minute. But I light direct test, I counted 0 minute and 60 minute. And here the graphical presentation for the 0 minute and 60 minute. We can observe that a seed for the extract of seed shows the more prominent activity, 220. Than comparison to footfall. This is my conclusion, and my future expect. From this research work, we found that footfall possesses a higher phenolic and flavonoid content, and along with excellent antioxidant activity compared to the seed extract, as is evidence of BIIHR activity, 𝐕𝐷𝐻 radicase scavenge activity, and peroxide activity. Furthermore, we observed that analgesic activity footfall showed a more prominent activity. This is like pain sensation in both hot plate method and acetic acid and standard analgesic activity. On the other hand, more prominent and sedative anxiety activity was found for seed extract in all experiment model. This finding highlight that therapeutic potential of footfall seeds of 𝐎𝐫𝐬𝐴 more prominent than any other parts like footfall. My future perspective on this research is that evaluate the phytochemical more potential, correlate the Amora genus, other Amora genus remain. I on this research I evaluate the one species like Amora rohituka. Another plant was Amora chittaganga. That also I work on that. I didn't mention here. I only describe about the Amora rohituka. My finding information more in vivo approach and compared to the data in vitro in vivo approach, we can use this finding. And we can also explore molecular docking through the compound in the commercial software like other way. And this is about my referencing. Thank you for your attention. And if you have any question, you can ask me.
ধন্যবাদ কাজী জান্নাতুন নাইমকে।